Hello, my name is William Reddy, and along with the contributions from Emerson De Silva, Tom Kovala, Mike Camito, and Jerusa Senhorinho, we will be presenting how green microalgae cultivated under extreme environments exhibit anti-cancer activity and selective effects against breast cancer cell line. Disclosure of affiliations, financial and in-kind of support. Grants/research support, Natural Sciences and Engineering Research Council of Canada, NHRC, and Canadian Bureau of International Education. This session programmers receive financial support from NHRC and Canadian Bureau of International Education in the form of an educational grant. Introduction. Breast cancer was the most diagnosed and deadly cancer in women worldwide in 2022. Despite ongoing research into new compounds, challenges such as chemotherapeutic failure, acquired resistance, and drug toxicity remain significant in clinical settings. Therefore, developing new compounds against breast cancer is crucial. Studies have highlighted natural sources such as microalgae as potential sources of anti-cancer compounds. Environmental stressors, including temperature, pH, nutrient availability, can influence the metabolite production of green microalgae. Materials and methods. Samples collection and isolation. Samples were collected from northern Ontario water bodies, where low pH and/or high metal levels are likely. Once the algae flourished, 20 microliters was transferred and streaked onto bulbs, basal, medium agar, and left under light for 3 to 4 weeks. Samples were then subcultures until monocultures were obtained, and the resulting algae strains were previously identified through morphologic identification and DNA sequencing. Preparation of extracts. 6 microalgae strains were used in this study. Sample 5, S5, and P918 are Coxomyxus species. LL1, Sendemesis species. LL2A, CC, and CR are Chlamydomonas species. The strains were grown for 28 days in bulbs, basal, medium, with pH 7 acting as a control and 5 under 16-hour and 8-hour light-dark cycles at 25 degrees Celsius, and were kept under continuously agitated at 150 RPM. Then, microalgae was harvested and methylonic extraction was performed. This final crude extract was then weighed, and DMSO, dimethyl sulfur oxide, was added to obtain a stock solution of 50 mg per ml. Figure 1. Wild green microalgae from various sampled northern Ontario water bodies, light microscopy 40x. Results. Figure 2. Anti-cancer activity of microalgae extracts against MCF7 cells after 24 hours of treatment. After attachment, MCF7 cell lines were treated with A, 150, and B, 200 micrograms per ml of microalgae extracts cultivated at pH 7, seen in the gray bars, and black at pH 5. Non-treated cells were set at 100%. Data was presented as a plus-minus standard deviation of the triplets. Statistical analysis performed with a t-test. Significant difference are marked with an asterisk, p-value less than 0.05. Figure 3. Cytotoxic activity of microalgae extracts against MCF10A non-cancerous cell lines after 24 hours of treatment. After attachment, MCF10A cells were treated with A, 150, and B, 200 micrograms per ml of microalgae extracts cultivated at pH 7, the gray bars, and 5, the black bars. Non-treated cells were set at 100%. Data was presented as a plus-minus standard deviation of the triplets. Statistical analysis was performed with a t-test. Significant differences are marked with an asterisk, p-value less than 0.05. Outcomes. Microalgae extracts exhibited significant anti-cancer activity against the MCF7 breast cancer cell line. The extract showed selective effects against MCF7 cells, since the viability of MCF10A non-cancer cell lines remained around 100%. The CC and P918 strains cultivated at low pH showed higher cytotoxic activity against MCF7 cells than those cultivated at neutral pH. The S5 strain at lower pH conditions showed enhanced cellular viability when compared to the same strain at neutral pH against the non-cancer cell lines seen in Figure 3A and B. Conclusion. The green microalgae extracts exhibited strong anti-cancer activity and selective effects, particularly from strains cultivated at low pH. These findings suggest that extreme environments can modulate metabolite production, highlighting green microalgae as a promising source of new pharmacological compounds.